Hey guys, welcome to Biomics Hub. Today we are going to discuss about the G1-S phase transition. So in the last video, we have already discussed about the basics of the cell cycle regulation. That means what is a cell cycle, how it is regulated, what are the proteins involved during the regulation we have already discussed. So if you have not watched that video, please do watch and come to this video. So let us see the overview how the cell cycle is regulated. So when a cell is undergoing division, it is going through various phases of cell cycle like G1 phase, S phase, G2 phase and M phase. During this phase, various types of cyclines as well as CDKs are involved. So the first is a G1-S phase CDK which is used in the G1 phase or the G1-S phase. So what is the work of the G1-S phase CDKs? The first work is that it inactivates the CDH1. What was the CDH1? The CDH1 was a activator of this APC. When CDH1 is combining or it is binding with the APC, then only this APC was activated. If not, it is not activated. So where this APC is used? This APC is used during the mitosis phase. Okay, when the cell is ready for the division, then that this APC is working. So it inactivates given as phase CDK. What is does it? It inactivates the CDH1. The next work of the G1-S phase CDK is that it activates expression of S phase cyclin CDK component. Whatever the cyclin as well as the CDKs which will be worked in the S phase, here that components will be activated. The next work is that it phosphorylates the S phase inhibitor. What happens in the S phase CDK? There is an inhibitor present. That means it is inhibiting the work of the S phase. The G1-S phase, what is does it phosphorylates this inhibitor. When the cell enters into the S phase, there is the removal of this inhibitor. How it is removed? The SCF, which is known as the SCF proteasome, it is having a ubiquitination property. It degrades the phosphorylate S phase CDK inhibitor. So what happens? The G1-S phase CDK was phosphorylating this inhibitor. That means now this inhibitor was activated. That means it is inhibiting the S phase CDK. When the cell enters into the S phase, there is a proteasome complex which is known as SCF. It degrades this inhibitor in the S phase. So now the SCDK is activated now. The inhibitor is not inhibiting the S phase. S phase CDK. So if this S phase CDK is now active, what is this? It help in the activation of the pre-replication complex by which the DNA is replicated. Okay. So now this S phase was starting from the late G2 phase and it is active till the late G1 phase and it is active till the G2 phase. Next come the mitotic CDKs, or we can also call it as MCDK. What is this is the CDC25 phosphatase activates mitotic CDKs which activates early mitotic events. So with the help of this diagram, we will see what it is going to say. So this V1, there is a kinase which is known as V1 kinase. This V1 kinase which is present, it is a inhibitor kinase. What is does it? As it is a kinase, it phosphorylates the CDK. If this phosphorylation has taken place by V1, then the CDK is inactive. So this inhibitor phosphate should be removed out. It is removed by the CDC25 phosphatase. So let's come to this slide. So the M mitotic CDK which is present, like this S phase CDK was having a inhibitor, this mitotic CDK is also inactivated. So the inactivation is removed by the CDC25 phosphatase. Now here we can see that what happens in the metaphase, the chromosomes which are present, they are present in the metaphasic plane. Now the cell is ready to divide. So when it is ready to divide, or when the chromosomes now can separate into two parts, the APC CDC20 proteasome degrades securin. So this was a chromosome. So if this is the chromosome, this chromosome is attached by a protein known as securin. Okay, what does APC20 does? It degrades this securin. If the securin is degraded, there is another component which is known as separase. It helps in pulling down this two chromosome into the two poles. So now this in the anaphase, this is the anaphase promoting complex. So during the anaphase, the chromosomes, they will be pulled towards the two different poles. Now what happens? Phosphatase activates CDH1 and APC CDH1 proteasome degrades mitotic cycline. So what does this mean? So here there is a phosphatase. So the CDH1, when in the CDH1 the phosphate is present, then it cannot bind with the APC. So in this case it is inactivated. It is inactivated during the S phase and the mitotic cyclines are stable during this phase. But when this APC needs to be activated, there is one phosphatase which is known as CDC14 phosphatase. What does it? It removes the phosphate from the CDL. The three phosphates which are present, it will be removed and the APC will be activated. When this APC is activated, it degrades the mitotic cycline. Then the two cells will be divided. Then the cell will be divided into two daughter cells. This is all the overview of this cell cycle. So let us see how the G1 phase transition is taking place. So as we have discussed that there are various types of cyclines as well as CDKs involved during this G1, G2, S and M phase. So for the G1 phase, the cycline which was involved is cycline D. So how it is activated? How the cycline D is activating the CDK? We already know that the CDK which is present, it is a cyclin dependent kinase. That means it depends upon the cyclin for its activation. The CDK which is present now, it is in the inactivation form. There is a T loop in the CDK which is having a activation site within it, and when it is in the inactive state, it is not exposing the activation site. When the cyclin D is activated or when the cyclin D is synthesized, it will come and bind with this CDK and it will phosphorate and it will change the structure of this T loop, by which the T loop will expose its active site. Then there is a activating kinase which is known as CAC or CDK activating kinase. It will phosphorylate the active site of the T loop and now the CDK cyclin complex is activated. In this way, the cyclin and CDK of the G1 phase is activated. So this is the activation case. We have to see how it is destructed when it will enter into some other phases. It needs to be deactivated, or if there is some problem, that means the G1 to S phase transition will not take place. During that case, the cyclin needs to be degraded. So how it is degraded? Let us see that. So this is degraded by a complex which is known as SCF. And the SCF which is present, it is a E3-ubiquitin ligase. Okay. So that is the inhibitor which is known as CKI. Okay. So how it is regulating the cycle? The CKI which is present, it is bounded by the CDK complex, cyclin and CDK complex, and it is inactivating the cyclin and CDK complex. And the E3-ubiquitin ligase, which is the SCF, it is phosphorylating or it is adding the polyubiquitination chain for its degradation. In this way, the CKI, which was negatively regulating the cyclin and the CDK, they are deactivated. So this is a E3-ubiquitin ligase, which is SCF, which is a E3-ubiquitin ligase. It is controlling this. There are some other components like CKIs. So one of the example of CKI is also P27. So the CKI which is present, it can be of CIP or KIP. So what is CIP? CIP means cyclin interacting protein kinase, or KIP means kinase interacting protein kinase, or only protein. So the examples of CIP or KIP is P21, P27, P57. So this inhibits the A cyclin or CDK2. The other CKI which is present is the ink. It is from ink 4 family. So the example of ink 4 is P16, P15, P18, P19. What does it? It inhibits the CDK4 or 6 and it inhibits the cyclin D. Okay. So here we are having a active cyclin CDK complex. The P27 which is present, it inactivates when it come and bind to the cyclin CDK complex and it is inactivated. In this way, the G1-S cyclins as well as the CDKs are regulated. Discuss about the G1-S phase transition. For the G1-S phase transition, it requires a growth factor. This growth factor is working as a ligand. This is working as a ligand and it activates or it forms a dimer of RTK, that is receptor tyrosine kinase. It forms a dimer. Then this dimer activates the RAS-MAPK pathway, RAS-MAPK pathway, or we can also tell it as that RAS-ERK pathway, RAF-ERK pathway. After it has synthesized this, it will enter into the nucleus. It enters into the nucleus. It activates the RAS, RAF, ERK or MEK pathway and this enters into the nucleus. After it has entered into the nucleus, it activates some of the transcription factors. The transcription factors are CMYC, FOS and Ju. So as these are the transcription factors, it will help in the synthesis of some other genes. So the genes here is the cyclin D. Cyclin D. These transcription factors that are the CMYC, FOS, Ju, it binds in the promoter. It binds in the promoter of cyclin D and help in the synthesis of cyclin D as well as CDK4 and CDK6. So what happens when there is no growth factor? If there is no growth factor, it will not synthesize the CDK4, 6 as well as cyclin D. Due to which there is a restriction point. What happens when there is a restriction point? The cell cannot enter into the S phase from the G1 phase. Now after the cyclin D, CDK4 and 6 has been activated, what happens? What is the work of this maturation promoting factors? So the work is that there is another factor which is known as RB or retinoblastoma. This CDK4, CDK6 and cyclin D help in the phosphorylation of this RB or retinoblastoma. So how does RB work? This RB, when it is in the phosphorylated form, if it is phosphorylated, it is in the inactive form. But when this RB is not phosphorylated, it is in the active form, right? So what is the work? What is this if it is active? That is another one transcription factors that is known as E2F. This RB binds with this E2F and it does not allow the work of E2F to perform. That means when this RB is binding with this E2F, the transcription is repressed in this case. But when CDK4, CDK6 and cyclin D are present, it phosphorylates the RB. Okay. But remember one thing, that when only CDK4 is phosphorylating, it cannot be active or it cannot be inactive because we require inactive. If it is inactive, it will leave the E2F. So for that, the CDK4 as well as CDK6 will phosphorylate along with cyclin D. So now it is phosphorylated. That means it is not in its active condition. It is inactivated now. If it is inactivated, it cannot bind with this E2F. This transcription factors which was present, it will be activated now and it will bind with this promoter region and it will do its work. Okay, when this E2F is activated, what it does? It binds with one promoter of cyclin E and cyclin A. Okay, this cyclin E and cyclin A was responsible for the G1-S phase transition as we have already discussed in our first video as well as in this video. So here the E2F is working as a transcription factor for the synthesis of cyclin E as well as cyclin A. With it, CDK2 is also activated. So what does this work? The cyclin cyclin E and CDK2. They are responsible for the G1-S phase transition. It is responsible for G1-S phase transition. Now, let us see the work of cyclin A. The cyclin A as well as CDK2, what is this? It does the centriole duplication. Centriole duplication has started in this phase when there is the activation of cyclin E, cyclin A and CDK2. Okay, let us now again see how this RB is working. This E2F and RB, when this RB was capturing this E2F, the gene was repressed. That means the work of the activation of the cyclin E, A and CDK2 is not being done here. When CDK and cyclin and CDK comes and bind with this RB, what is this? It phosphorylates and there is a dissociation of E2F. This RB, you can see here that when there is phosphorylation of this RB, the E2F free now and it can bind with the promoter region for the synthesis of cyclin E, A and CDK2. So now this E2F is binding here and that is transcription and that is synthesis of cyclin E, cyclin E, cyclin A and CDK2. Due to which there is a transition from G1-S phase. So now let us see how another factor that is P27 is regulating. This is a progression through restriction point and entry into S phase is immediately mediated by the activation of CDK2 cyclin E complex as we have discussed earlier. But there is another factor which is known as P27. What does it? It binds with the CDK2 and cyclin E and it does not allow the further synthesis. Or we can tell that during the restriction point, this P27 blocks the MPF to work. So when there is synthesis of the cyclin E, there is a repression of P27. So what the cyclin E does? It helps in the phosphorylation of P27. Due to which there is a degradation of P27. Now the CDK2 and cyclin E is free. So it activates all the things which are required in the S phase transition. So let us see again with this diagram how this G1-S phase transition is taking place. So here we are getting a mitosin or mitosin factor which is a growth factor. It is binding with the receptor. It is activating the RAS-MAP kinase and it is entering into the nucleus. When it is entering into the nucleus, it is activating some other factors for cyclin D as well as CDK4 as well as CDK6. So here that is a complex form. What this complex does is it activates or it inactivates, it phosphorylates the RB which is binding with the E2F and inactivating the E2F. When the cyclin or the CDK, when the cyclin CDK complex helps in the activation of this RB now is in the inactive form. Here the RB is in the inactivated form. And this E2F which was present, it is working as a transcription factor and the S phase transition is taking place due to which there is synthesis of cyclin E as well as cyclin A. Along with it, CDK2 is also activated which help in the formation of some of the transcription or some of the cyclins which is known as cyclin E and A which is responsible for G1-S phase transition and due to this there is a synthesis of DNA. This is how the G1-S phase transition works. So in the next video we will discuss about the M phase or the mitotic phase, how the transition is taking place in the mitotic phase.